All right, well, thanks. And thanks everybody for joining today's webinar. My name is Scott Berry, a biostatistician here at Berry Consultants. And on behalf of the ICTR and NHLBI, going to spend an hour here talking about historical borrowing in clinical trials. So George Santayana famously said, "Those who cannot remember the past are condemned to repeat it," which he probably wasn't talking about clinical trials, but we can think about this within clinical trials, that if previous data that's been generated is ignored in trials, are we just repeating things that we could use within the science of clinical trials? So the potential here is that by using historical data, we're remembering the past, but we also avoid recollecting it. In a clinical trial, we might be able to randomize fewer patients to a control and use historical data on that control. It enables us to run smaller trials, faster trials, stronger trials by using that previous data. This is, of course, the potential that we would all like to tap into with the historical data. The concern in here is that what if the future is different from the past, and we're using historical data and it's not actually appropriately measuring what the world is now, and our inferences are off. So we can draw wrong conclusions in trials by using historical data. So we have this great potential, but also this concern. So I'm going to spend a good bit of time today in today's discussion talking about both of these. So we're going to discuss the use of historical data in the analysis of clinical trials. We need to understand the risks and benefits of doing that. We talked a little bit about the potential benefits. What are some of these risks? And can we mitigate those risks so that we can use this historical data with less worries that we're drawing incorrect conclusions from these trials? So this is what we'll focus on. So historical data, what I mean by this is that think about where we are in our current state, that this is data that's been collected before and we know the data. The data is known. This isn't a situation where we're proposing a phase 2-3 trial and we're going to use the phase 2 trial in the future and we haven't collected it yet. This is known data from the past. We are now designing a future trial, and we're interested in using that historical data. That's what I mean by historical data. Now, there can be many forms of this historical data. We use nuisance parameters all the time. We might be building a model that we use to analyze, and implicitly we've sort of used some of that data to build our models, but it's not explicitly used in the analysis. There can be longitudinal data that might even be used. Those components are used, variance estimators. I'm not really worried about these. They can have really minor impacts on things, but let's ignore those types and let's focus on two types of historical data. The most of what I'll talk about today is historical data on a control arm. So we're interested in a new experimental arm and we want to compare it to a control, and we have previous data on the control. The other one, and I'll talk about this briefly, it has similar issues where we actually have historical measurements on the treatment effect of our new experimental therapy. And we're bringing in an effect size from historical data that has different ramifications within these trials. So I'll talk about that briefly as well. So the setup is I'm interested in the comparison in a new trial between experimental agent A, experimental arm A, and a control C. It's not unusual that we have previous data on the behavior of C. It's now perhaps the standard of care, and there have been previous studies that have investigated C, and we have that data. We have no previous data in this part on the behavior of experimental arm A. So this is new, and we want to compare it to C. So what are the issues with using data for C, and how do we use that data for C in this? There's multiple ways in which we could explicitly use it in the analysis at the end of this trial. So let's look at a simple example. So we have historical data that the control rate has a success rate of 30%. So we're measuring our therapies on a dichotomous outcome of success. And historically, we've seen the control have a 30% success rate in a previous trials. We want to run a new trial with 20 patients on the new trial. One way to use that historical data is run a single arm trial to compare to the 30% success rate. In some way, this is the strongest level of historical borrowing, is we actually assume we know this success rate for control C. So we're not going to use a prior for it. We're going to assume we know it's 30%. We're going to run a new trial, single arm for A, and it's going to compare to 30%. So from this simple example, some of the aspects of historical borrowing come out. So in this single arm trial, we're getting 20 observations on A. We're interested in the response rate PA for that. We're going to test whether that's greater than 30%. If we reject the null, we're going to claim that the success rate's greater than 30%. Why we're doing that is because the 30% was estimated to be the rate of success on control. Notice you could, of course, do a two-arm trial where you have the control rate to compare to, but here we're plugging in 30%. For a sample size of 20, we can calculate that we would reject the null hypothesis if we observe 10 or more successes for A in this new clinical trial. That rule, that test, has a 4.8% type 1 error rate. So if in truth there's a 30% success rate for A, the chance we get 10 out of 20 or better is 4.8%. If PA has a 60% success rate, we can talk about power. The likelihood of rejecting the null is 87% in this scenario. This is a standard calculation you'll see done for a trial like this. We've got a type 1 error control. We've got 87% power. We've actually used the historical data to construct the 30%. If this test has any relevance whatsoever, it's because we think the control would have this 30% success rate. The issue is that 30% might not be right. And what does that mean, that 30% might not be right? The data clearly showed that 30% historically was what C did. But what if in this new trial we actually enrolled control and it had a 50% success rate? That could be due to different inclusion-exclusion criteria. It could be due to time, that patients are now healthier. We've enrolled different patients, but that if there was a control rate, it would have a 50% success rate. Then PA is really 50%, and it's going to have an elevated type 1 error under the assumption that PC is 50%. So it really comes back to this question of what is type 1 error? You can go back mathematically and think about the pure hypothesis of 30%, but what we're really doing is saying, is A superior to C? And now we could be making type 1 errors if C would actually be different than 30% in the new trial. So let's take a look at what that looks like. So the x-axis on this graph is the rate that control would have in the new trial. We know historically it was 30%, but in the new trial, what rate will control have? It might be that it's exactly this 30% rate. That's all well and good, and that things haven't changed in our new trial or over time. If that's the rate, what I've graphed here is the type 1 error rate for the trial, assuming our new experimental agent has the same rate as control, and this control rate can change. So if it's 30%, that's the 4.7% that we mentioned before. But under the example I gave, where the new control is actually 50% if we would enroll it, we have the probability of 58% of rejecting the null and saying A is better than C. That's a type 1 error because in reality, they're no different. Control is 50% and active is 50%. But we reject the null against 30%, which is our estimate of the control, and we have no new data on the control. So that trial has a 57% type 1 error rate compared to what this new control rate would be. Now, we could instead, if that's a concern, we could take those same 20 patients, we could ignore the previous data. We're running a new trial and only data from the new trial is going into it. And as I described, we can instead compare the probability of success on A to the probability of success on C in this trial. We're going to enroll 10 on each of the arms with same sample size of 20 in this new trial. And there's actually a discreteness issue of getting the type 1 error rate. So you can use an alpha of 0.1 in this setting to actually have a true control type 1 error less than 5%. So I'll show you what this looks like. So I've taken the same graph that we had before. That's the type 1 error rate where we assume we know the rate is 30% on control. We're borrowing the previous number. Actually, we're borrowing it to an extent that we assume we know it exactly. It's not even using the data with uncertainty. That has about a 4.8% type 1 error rate if 30% is true. The orange line here is if we were to run a new trial and do 10 to 10 randomization. Our type 1 error is controlled over this range and has a very similar type 1 error if in fact 30% is right. The negative of that is that we've thrown away a great deal of information on the control, that 30% rate. So if we look at the power, assuming that the active is 60%, if the control rate really were 30%, our new trial that does 10 versus 10 randomization has barely over 40% power to show that A is better than C. The trial that enrolled all 20 of those patients on the active arm A has 87% power. So we get enormous power advantages in using that historical information. And as you can see, we improve power by over 40% in this scenario. Now, I'm going back a slide. The risk of that is if we're wrong on the 30%, we can have enormous type 1 error rates. And you can see if we have drift from the historical data to now of 40%, so it's increased 10%, we've now got over a 20% type 1 error rate. That's the risk, that's the negative. The potential and the power is this enormous increase in power in this trial in being able to enroll everybody to A and comparing to 30%. Increases our power by this much. It also means we get to enroll everybody to the new experimental arm A, and we don't enroll any new C patients. We don't repeat it as Santiana described on the first slide. Now, there are alternatives here. We don't have to go to the extreme that we know C is 30%. We might not have to go to the extreme that we ignore the historical rate entirely. What if we incorporate that data? So let's assume, for simplicity, that we had 10 historical observations on C, and 3 out of 10 were successful. This is that 30% rate. Under this circumstance, it would seem overly confident to say we know the rate is 30%, for example. But what we're going to do is we're going to do a Bayesian analysis where we include that data, that 3 successes and 7 failures in the trial. This is essentially pooling that data with the control arm from the new trial and using 100% of the weight of those 10 observations. What we can also do is change the randomization rates. We can randomize 15 on active A and 5 on the control C, because we're bringing forward 10 historical observations on C, which is going to result in 15 on each arm. So we're enrolling a trial of 20 patients, but now it has the strength of 30 patients because we're using the historical data. On this, I've graphed that the same type 1 error for this and the same power for this, comparing to the 2 previous alternatives. So the pooling is in purple here. That shows the type 1 error. So rather than assuming we know the rate is 30%, we're using 3 observations out of 10. Notice our type 1 error is actually decreased. We make better decisions by using that 3 out of 10 when 3 out of 10 is right. So it's this we're going to see this over and over throughout the slides and the ramification of using historical data. If the historical data you're using is right, it's great. If it's wrong, it can hurt you. And we'll talk about ways maybe we can learn if in fact it's right during the course of it. The pooling here improves type 1 error by using that data relative to not using it, no borrowing at all. It doesn't have quite the power improvement that we got with 30%, assuming 30% was right and putting all 20 patients on active. Now we have 30 total patients in this setting, 15 on each arm. So it's the power, the pooling, the type 1 error doesn't have nearly the negative effect that assuming 30% is right has in this setting. If the true rate is 50%, we don't have this 58% type 1 error we talked about. Our type 1 error is only 13% in that setting. So we've got much smaller type 1 errors in this setting. And again, decreased type 1 error if in fact 30% is right. The power ramifications, when in fact the new therapy has a 60% success rate, you can see we do quite a bit better than not using that previous historical data. We're running a trial that if 30% is correct, our power goes from this 41% to now almost 70%. It's not all the way up to 87% that we had with the single arm trial. Notice I recalibrated things a little bit to make a fair comparison on power because we had decreased type 1 error. So I just raised the threshold so it had similar type 1 errors to the other 2 to show what its power would be in comparison to no borrowing or a single arm trial. So there's a number of things that come out of this that I've sort of been pounding on here, is that if the historical data is right, we do really, really well in the setting. If it's wrong and significantly wrong, we can really make quite negative conclusions in the trial. So it brings about the somewhat obvious question, can we learn from the data that we collect which of these methods we should use? If we were to observe control data near 30%, maybe we use all the data. I don't think we go to the extreme that say, oh, we saw 30% out of 10 patients, 3 out of 10 in the new trial that we assume we know it's 30%, but maybe we'll fully borrow the previous data in that setting. Or if we're close to 30%, we want to borrow the previous data because it looks like actually our new control is similar to the old control and the historical is right in some sense. If we start to observe data on the control that differs from 30%, we start to worry that we're now measuring a different control and we want to discount the historical data. So I'll talk very briefly. In a somewhat obvious thing that might come to mind is test and pool. Let's do a significance test to see whether it's the same as 30%. If we reject it, we'll discount, we'll throw all the data away. Otherwise, we'll use all the data. It's a really all or nothing type approach to dynamically borrowing from the historical data. Bayesian hierarchical modeling is another way to do this, and I'll spend some time using demonstrating the Bayesian hierarchical modeling. So I'm also going to change the example a little bit, and I want to highlight this paper. It's a really very well done paper. It was a working group from the Bayesian working group. Kurt Veely was the lead author on this, so I'll refer to this as the Veely paper. There's an example that's carried through and a number of approaches are looked at. So this is a great resource to go to and look, and I'll borrow some of the results from that paper. So a very similar setting where we're going to run a new trial of 400 patients. We're going to do one-to-one randomization between A and C in the new trial. If we're going to do this borrowing, of course, we probably are going to do a different randomization between A and C. But we can demonstrate the ideas within this equal randomization in the new trial. The historical data has 65 out of 100 successes for control. So we've seen a 65% success rate historically, and we're interested in seeing if we can demonstrate that A is better than C. The question is, what do we do with the 100 patients? What do we do with that 65% rate? So Bayesian hierarchical modeling, what it does is we model the variability between the sources of data. So we use a logistic transformation here where we model the log odds. We model the log odds of the historical control, which I've labeled with the sub-0. And then the new log odds in the new trial, I label it without the 0. So that's the newly collected data as gamma C. We model this with a hierarchical distribution. We use a normal distribution with mean mu and variance tau squared. What makes it hierarchical, and it's where the term comes from, is we model the mean and variance of that with their own distributions. Notice at the extreme of this, if tau squared were very large and we plugged in near infinity, there would be no borrowing between the two trials. This distribution would be flat and there'd be essentially no borrowing between the two. If tau squared is 0, it corresponds to pooling the results from the two trials. So instead, we put a prior distribution on tau squared, and I've demonstrated one of them. I'll show you several priors on tau squared, but this prior is one that works pretty well in this scenario, an inverse gamma distribution with parameters 0.001 and 0.001. What's going to happen here is the model is going to be able to look at the similarity of the control rates. If they're similar, the posterior for tau squared will be small and more will be borrowed in this setting. The prior for mu is also hierarchically, this is the second stage of the prior, but that plays a relatively small role in the conclusions of this. It's tau squared that plays an important role in the analysis. So here's a demonstration, and this is where the term dynamic borrowing comes from in this setting. So I'll spend a little bit of time and explain what's going on in these graphs. On the left is the expected number of borrowed observations from the 100. The different methods here in red is just pooling, and red is pooling over here to the right as well. It means we add 100 patients to the trial inference. And the y-axis here is the true rate of control in the future trial. This is where if 65% is that rate, that's measuring exactly the historical data. So in that setting, this is the amount of data that's borrowed is 100. Well, if it turns out that we have something different and it's 80, pooling still borrows 100. This is what represents static, that regardless of the data in the next trial, we borrow 100 patients. Ignoring those 100 patients is the orange line here at the bottom, where regardless of the data, even if we observe exactly 65%, we ignore that data. The blue lines are different power priors where you downweight the 100 to be equivalent to 80, 60, 40, or 20. And again, it's static. A power prior represents static borrowing in this setting. If we were to use 40% weight, we use 40% regardless of the future data. Hence the term static. Over here to the right is dynamic borrowing. These are, the red and the orange are the same full borrowing, pooling, and no borrowing in orange. And then each of these curves represents the average number of borrowed observations using the Bayesian hierarchical model. Each one of these corresponds to different prior distributions for that. And again, you can represent a prior distribution that makes this model look like ignoring or look like we want to borrow quite a bit. What the dynamic part of this is that for each one of these, we borrow more data when the control in the new trial is near the 65%. As the data gets farther away from 65%, we borrow less. This is the dynamic aspect of it, which has a really nice scientific appeal in this particular setting. I'll focus on the prior, the 0.001, 0.001 is this solid green prior to show how that works. So here's now a comparison. This is, again, the type 1 error graph that we've seen before for this example where the historical data is known to have a 65% success rate. This is the type 1 error. Now, we worry about the type 1 error in particular because it gets increased as the control rate in the new trial is bigger than 65. So let's review what this 0.75 means. That means that in the new trial, the success rate for control is 75%. And since this is the null, the success rate for the experimental A is also 75%. So there's no difference in the two treatments. The type 1 error of doing full borrowing, full pooling of the previous 100 patients is about 10%. Remember, in this trial, we have 400 new patients and 100 borrowed. So we don't have the extraordinary increases. Now, if it was 80%, we're looking at about 20% type 1 error of just fully pooling the previous data. The blue line represents the type 1 error of downweighting the previous 100 patients to have the equivalent of 40 patients. It's a power prior with a weight of 40%. Now, the type 1 error at exactly 65 is smallest with the pooling. It's a little bit larger for the power prior in that setting, but it has not as negative, not as bad type 1 error if we have positive drift in both of the arms. Notice the Bayesian hierarchical model, its type 1 error starts to decrease as that control rate increases. So as that rate increases, it's discounting the data and it's starting to look more like no borrowing. And we limit the size of the type 1 error of the dynamic approaches, where the power prior is going to continue to increase and pooling is going to continue to increase because we blindly use the data regardless of the new control. I wanted to show you test and pool. Test and pool is actually a dynamic procedure. Notice what it does is it mirrors the pooling up until the point where at these levels we tend to start to reject that the new control is like the old control, and it starts to drop down and look like no borrowing. So it actually has a dynamic behavior to it as well as the Bayesian hierarchical modeling. I haven't said much about it, but one of the other aspects of this is what if we have a decrease in the rate? The new trial control rate under C would actually be less than 65%. We're enrolling a different kind of patient. In these settings, we have type 1 error deflation. It's very much deflated for pooling within that setting because we're using data on the control that is better, and the new active arm has a smaller rate. It's harder for it to separate. This graph is the power graph, assuming that A has a 15% absolute improvement over the control. Again, there are a number of lines looking on here, and some of the messages are similar to what we were looking at before. Notice if 65% is right, the Bayesian hierarchical model has very similar power to the pooling method in this setting. It does like pooling. It does really quite well. But as that control rate is increased, this starts to decrease its power and it starts to look more like no borrowing. It also has that advantage as the control rate moves to the left that we're enrolling a less healthy population. It starts to look more like no borrowing as that control rate gets lower. That's what we would want it to do. Ideally, we would want the trial to look like no borrowing away from 65% and look like pooling at 65%. And that's the way these dynamic procedures work. That's exactly what the Bayesian hierarchical modeling does in this setting. Compared to the pooling, that again, it blindly borrows no matter what. So does the power prior. It just borrows less. Again, a static approach. So the negatives of that, the historical is measure something different, is exaggerated in the static approaches. It is mitigated to some extent with the dynamic approaches. So different static modeling choices. Pooling, you just blindly pool the data. You can use power priors, which discount the past data. But again, these are it's a static approach, and I walk through what that meant in terms of regardless of the new data, we're going to use some percent of the data. No borrowing is a static approach. Regardless of the new data, we're going to ignore the previous data. It's a choice in the setting. A single arm trial, a lot of times that 30%, we were running a test comparing to 30%, that's referred to as an objective performance criteria. We're trying to beat a number, and that number comes from the historical data. If we're going to run a trial comparing to a number, that's even more extreme than pooling. It's assuming we know the answer in this setting. These are all static approaches to using historical controls. Dynamic approaches, you saw test and then pool, is a dynamic approach. Hierarchical modeling is a dynamic approach, and we walk through that a little bit. That's a particularly good choice if we have multiple studies. I just walked through an example where there was a single historical trial. What if we had multiple studies? It really becomes a meta-analysis to estimate the true control rate. And Bayesian hierarchical modeling, or hierarchical modeling in general, you could do random effects modeling, works really well in those settings. It's a very natural choice. Cluster modeling advances the hierarchical modeling to allow multiple numbers of distributions. If you have a lot of trials and concerns that there are clustering of control effects, this can be quite powerful. It's advancing the Bayesian hierarchical modeling to multiple hierarchies. Mixture priors are where you model the new control as some probability it's exactly equal to the old control, and then some probability it's measuring something else. This is a mixture of a continuous distribution for that control and a discrete point at them being exactly equal. This is dynamic, and it works really quite well if you only have a single historical data set. You have the difficulty of creating the prior distribution for tau squared within the Bayesian hierarchical model, and you don't have that in the mixture model. You, of course, have what probability do we put on exactly equal in that, but sometimes this can be more straightforward. Commensurate priors are another methodology for trying to extend the power priors to have dynamic borrowing by modeling the power itself potentially dynamically. Now, I'll touch on briefly historical controls. Using historical controls inflates type 1 error. You can construct a new scenario where it inflates. There's no way around that. The only way around it is to ignore it. The question is, when do we care? We may spend a lot of time addressing, do we think that the new data is going to be different? We could compare demographics. What are the inclusion-exclusion criteria? Comparing patients. How much time has gone on between them? Have we added new therapies? Did the data we got for C potentially come out of publication bias, that we're actually only seeing the better data for C? The concern then is actually the rate's smaller. Are there other biases? Such as multiple phase 2s running and we only hear the results of positive phase 2. So that's a very much an important thing to address. And in these settings, we may very much believe that we're measuring very similar things, and we're much more willing to borrow the controls in the sense that we're getting the very strong advantages and we're not worried about the risk of the drift that could potentially fool us. Again, ignoring historical data is a choice in this setting. Dynamic methods can very much mitigate these risks. So if it really does look like we're measuring something different, the modeling adjusts for that. What about borrowing effect sizes? So I'll move away from only where we have data on the control to a setting where we actually have data on the effect size. Not surprisingly, if you borrow positive data on the effect size from historical data, you inflate type 1 error. And you give yourself a leg up to reject that A is better because you're bringing in data that already shows A is better. You cannot include positive prior data and control type 1 error. They're opposite inferential quantities, and they don't play well together because type 1 error is purely on the sample space, and the prior data isn't in your sample space. So what does that mean? Let me show you a very simple example. So a single-arm trial where I'm trying to demonstrate whether pi is greater than 0.5. I'm going to run a 100-patient trial. And if I see data for X is binomial with 100 observations, pi is the probability of response. If the probability pi is greater than 0.5, given my data is greater than 95%, I'm going to say that's trial success. This is only using data in the new trial. This is equivalent to using a uniform prior for pi. So I'm going to use a uniform prior, beta 1,1. If at 100 patients, we can calculate for every one of these values which values of X we would reject the null. We would need to see 59 or more successes in order to meet that criterion and reject the null. That has a one-sided type 1 error rate of 4.4%. What does that mean? That means the probability, if the true rate is 50%, if we're flipping a fair coin, the probability of seeing 59 or more successes on 100 tosses is 4.4%. That's calculated only on the new data. It doesn't calculate from the previous data. There is no previous data in this example, but I'm going to add the previous data. But you can see how that type 1 error is calculated. Now, suppose on the same therapy, we have 10 patients from historical data, and 9 out of those 10 were successful. Let's assume we're not worried about a publication bias. This is the only data that's ever been calculated on this therapy. We could use a beta 9.2, which is updating the beta 1.1 based on 9 successes and 1 failure. So instead of the beta 1.1, we use a beta 10.2. We're starting with a leg up with good data. Now, in order to have a probability of 95% that we're better than 0.5, we only need to see 55 successes in the next 100 patients. Remember, it used to be 59, but because of the 9 successes and 1 failure, that's lowered to 55. And the probability of getting 55 or more, if 0.5 is the truth, is 18.4%. The type 1 error of the new experiment is 18.4%. Now, I could adjust this 95% threshold trying to balance the fact that I've used historical data. The only way to adjust that to get to the point where I control type 1 error below 5% is to get back to the 59. The 59 determines type 1 error, and so I could adjust my threshold, and this is an approach that people use, to 99.34% using my prior, my beta 10.2 prior. What that means is it's a way to reverse engineer that I get my 59 back. So if my type 1 error drives my threshold, it's essentially saying you need 59 out of 100. I don't care what historical data you have. It doesn't matter. 59 out of 100 is always the answer to type 1 error. Hence the conclusion that you can't use positive data and control type 1 error at the same level. You can't get its benefit and control type 1 error. The two things are at odds to one another. So this does not mean that we shouldn't use historical data. Reliable data that we're reasonably likely are free of biases. We believe it's all the data we have. It can demonstrate with that effect that the null is actually unlikely. We have a reasonable amount of data already that the null is unlikely. We don't care that under that null, we might get the wrong answer because we've already moved in the Bayesian continual learning away from the null hypothesis. You can read the Bayesian, the FDA draft guidance. They talk about using Bayesian analyses and the probability of incorrect conclusions. But again, if you approach this from type 1 error is what drives things, you lose all of the advantages of that data. So it's incredibly important we utilize that data. We have to understand and report the behavior of how we use it, prospectively define that, But we also have to alter our thinking that the inferential unit of the single trial and 2.5% is the be-all, end-all scientific standard. It's not, because it ignores the data. So these two things are very much at odds with each other. So I showed you Santiana's comment about those who don't learn from history are deemed to repeat it. I will present a quote from another famous philosopher, Rafiki, the baboon from Lion King, who said, "The past can hurt, but the way I see it, you can either run from it or learn from it." So yes, the past can cause errors. We can ignore it and run away from it and run solo trials ignoring it, or we can learn from it and do it well. So I like Rafiki's approach, and I think some of the methods here agree with that. So in this setting, so appreciate your attention, everybody, to this, and happy to turn it over and answer some questions.